of a Pakistani individual, the sequence and variant analysis of a Pakistani individual. And because of the geographical location of Pakistan, Pakistan is situated at the crossroad of South Asia, Central Asia, and the Middle East, with over 200 million population. And Pakistan is the sixth largest country of the world as far as population is concerned. Most of the Pakistani population has been characterized or classified into North Indian ancestry, which is genetically closer to the Middle East people, Central Asian and European people. In the South Asia, the region which was previously called as Indian subcontinent, which includes Pakistan, India, Bangladesh, Nepal, Sri Lanka, basically genetically has been divided into two types of people: North Indian ancestry people and South Indian ancestry people. Pakistani population, Pakistan basically is a part of the northern part of the Indian subcontinent and South Asia. Pakistan is situated on the northwestern part of the South Asia, and Pakistani population are basically part of the North Indian ancestry, and this North Indian ancestry is genetically closer to the people in the Middle East, in the Central Asia, and some eastern part at least of the Europe. Pakistan displays near maximal frequency of alleles that cause, cause or participate in a number of diseases, including diabetes and cardiovascular disorders. So Pakistani population exhibits considerable frequency of alleles or genetic variation that cause or participate in a number of genetic disorders, including diabetes and cardiovascular diseases. In 1000 Genome Project, samples, DNA, that is genomic DNA samples from Pakistan was included. Around 100 samples were included, 100 individuals' DNA samples were included in 1000 Genome Project. But the 1000 Genome Project, one of the that is disadvantage or the drawback of 1000 Genome Project is that it is the low coverage sequence data has been carried out in the has been included in the 1000 Genome Project dataset, which is 2 to 4x coverage data, which is called as low coverage data in genomics terminology. Therefore, data which was included in 1000 Genome Pakistani individuals data, genomic data, which was included in 1000 Genome Project dataset, might not represent the diverse ethnic groups living in Pakistan and the genetic diversity in this ethnically diverse population in Pakistan. So the genetic diversity of diverse population is yet to be characterized. Okay, we have a question, and I request that the question will be answered after the lecture, inshallah. Okay, so please keep your questions with you, and write down question with you. And after my talk, I will, inshallah, try to answer the questions. Okay. Next. This slide is showing two postulated routes out of Africa. So that is the African people, the home was the migrated from Africa into the other parts of in other continents of the world, other parts of the world. According to this postulated routes, one route was in the, you can say, the southern route out of Africa, this route, okay, and they from Africa to from southern part of Saudi Arabia, and then southern part of Iran, and then they entered into Pakistani part of the Indian subcontinent, and then through India they went towards the migrated towards the east. There is another route of out of Africa migration of Homo sapiens is this one, in which from the north, this is the northern route in which they migrated from Africa to the northern part of Middle East via that is the Syrian, from Syria they went to the northern part of Iran, and then they entered into the Pakistani part of the South Asia from the northern Pakistan. Anyway, so these two, both of these two postulated routes might be is possible, and some people might came from the northern route to Pakistan, entered in Pakistan, and some people came from the southern route into the into Pakistan. It was, you can say, more than 40,000 years before present. Okay, so we basically carried out the genome sequence of a Pakistani individual in order to find out the genome variation of Pakistani population, to start with the first individual. So what we did, we basically extracted DNA from the Pakistani individual, from the blood of that individual, because blood contains white blood cells, the leukocytes, and those white blood cells contain DNA, genomic DNA. So from blood, that DNA of a person can be easily extracted. And after that, our group carried out the library preparation of the DNA sequences, and after library preparation, the next generation DNA sequencing was carried out using the next generation DNA sequencer. So for that, we extracted 100 microgram of DNA from the blood of the Pakistani male individual, and processed, and the genome sequencing was carried out using HiSeq 2000 genome analyzer, manufactured by Illumina company, which is one of the major genome analyzer manufacturers in the world of NGS machines. Okay. Next, what we did, yes, the genome sequencing of Pakistani individual resulted in the short reads, resulted in the sequences. And because these sequences are around 100 base pair sequences, so we call them short read. So in order to analyze the sequences, we carried out the alignment of these short reads into with the alignment with the alignment with alignment with, excuse me, I could not see that. This is the procedure which I wanted to explain with you, that how we what type of bioinformatics analysis or computing, biocomputing was carried out after the sequencing was completed. The first thing was short read alignment. The short read alignment using the SOAP program, short oligonucleotide analysis program, the SOAP program. Okay. The SOAP programs were used for genome mapping and SNP analysis in order to find out the single nucleotide variants from the aligned data. And for that, we use the reference human genome dataset, which is called as HG19 dataset, which was used as reference for the short read alignment and identification of the SNP, short indels, and structural variation in the Pakistani genome. So we extracted the DNA, sequenced the DNA, extracted the DNA, sequenced the DNA, and then align the DNA with the reference sequence, reference human genome sequence, which is at that time, this version of human genome is called as HG19 version. Nowadays, there is another version is available, but at that time when we were doing this work, it was the latest human reference genome sequence, code name as HG19. So the HG19 sequences were aligned with the HG19 sequences were aligned with the Pakistani human genome sequence. Also analyzed our sequences with the DB-SNP, database SNP. I will discuss this database SNP. This is a database which contains all available reported single nucleotide variants from all over the world. Also, we compared our genome sequence, the Pakistani genome sequence, with 1,000 genome sequences, 1,000 genome sequence data set, also with ensemble genome data set and OIM data set, compared with the genome sequences of Pakistani individual. Okay, then we found out the characterized non-synonymous SNPs in Pakistani genome. Non-synonymous SNPs are those SNPs which cause that is change in the codon which code for amino acid. So in that is as a result of non-synonymous SNP, non-synonymous SNPs are non-synonymous SNPs are basically coding SNP, which code for amino acid in protein. So there are two type of coding SNPs, one called as non-synonymous SNPs, another is synonymous SNPs. Synonymous SNPs in which synonymous SNPs are those SNPs which basically, because of which codon is changed, but amino acid is not changed. As because we know that there are more than one codon code for one amino acid. Non-synonymous SNPs are those SNPs in which, because of the SNP, because of the change in the DNA sequence, codon is changed, and also the amino acid is also changed. So we basically analyzed the non-synonymous SNP, which we call during this presentation, we will call as the NS SNP, were screened for predicting damaging effects of missense mutation. Non-synonymous SNPs are also called as missense mutations. Missense mutations. So we wanted to see whether the non-synonymous SNP or missense SNP, whether they have damaging effects at protein level or not. They are amino acid is changed, but whether this change in change of amino acid is damaging the structure of protein, or whether it is damaging or harmful for the function of the protein. So for this, we used two computer programs. So a lot of bioinformatics. So a lot of computing is required in genomics. A lot of computing is required in genomic medicine. This we should keep in our mind. So for this, we use two programs. One program is called as SIFT. Another program is called as Polyfem, the version 2 of Polyfem. These programs are used use sequence-based and structure-based predictive feature to determine whether amino acid exchanges are likely to be damaging or not. So these programs predict whether the amino acid exchange is likely damaging or harmful in case of the structure of the protein or the function of the protein. So this was important to find out about the effect of DNA sequences, effect of mutation at not only at DNA level, genomic level, but as well as at protein level.